what you know about plants so far, it's pretty obvious that they have to respond to light. And it's not surprising that they have more than one way to do that. And so, of course, for a plant, being able to assess if they're in the sun or not, is it daytime or nighttime? Am I in full sun or am I being shaded? All of that information they're assessing constantly, and they have to respond appropriately in order to adapt and survive. And so the fancy word for that is, we've already talked about the phototropism idea, that they can, if you have unidirectional light shining on a plant coming through your apartment window, let's say, they will display that growth, that asymmetrical growth pattern in their stem tissue so that they can bend toward the light and orient their leaves to that incoming sunlight. So we've talked about that. They've actually, when you think about sunlight, if you think back to maybe middle school or elementary school, if you ever had a science teacher put light through a prism and then it would break into the rainbow colors. It's such a simple experiment, but so fun to see. So we know that sunlight is actually an array of different wavelengths of light quality. And so you might remember the red, orange, yellow, green, blue, indigo, violet, all the colors of the rainbow. And so they have deduced that plants can tell the difference. They know what red light is, they know what blue light is, they know what green light is, and they have preferences. So there's certain wavelengths of light that they prefer to absorb and make use of. Other wavelengths of light they don't care so much about, and they'll bounce that back off of them. And when we look at a plant, it bounces to our eyes, see it. So when a plant looks green to our eyes, it's primarily because while there's a ton of chlorophyll, they're not absorbing green quality of light, though. They're bouncing that away because they don't really work with the green wavelength of light. And so our eyes pick up green. And so when you look at this idea here with phototropism, so they actually were asking the question, okay, we know plants bend toward light coming through a window, let's say. Is it any sunlight, like all the wavelengths of sunlight, or the particular range of wavelength of sunlight energy? And so this picture is showing that where you see it peaking like that, that means that that's the wavelength of light that is causing, in this case, the phototropism to occur. So it's not just any old quality of light. They have to get access to blue light, which, as you see on this diagram, blue light is around 450 nanometers, that wavelength of light energy. And you can see as you work your way to the right of this picture, you start, you end up around red, and then far red is around 800 there. So red light has longer wavelengths of light. Blue light has shorter wavelengths, so it's higher energy. If it's a shorter wavelength, it's higher energy. If it's a longer wavelength, it's lower energy. And so here they were able to do the experiments to show that it's blue light that the plants are actually assessing, and then it's the blue quality of light that's sending the auxin over to the dark side of the stem and is causing that stimulation of growth on the dark side. So you get that asymmetrical growth, which leads to the asymmetrical bending. So they figured out that there's an actual protein called phototropin that is absorbing that blue light. And that's what's kicking off this whole, okay, we're getting this blue light from this direction. That means we need to send auxin over to the dark side. Okay, so that phototropin protein is very important to kick off that whole phototropism response that the plant is giving. Another thing that they figured out is that it's the blue quality of light that's actually helping to regulate the stomata, the guard cell movement, whether the guard cells should move away from each other and reveal the pore, or whether they should shut down over the pore. And so they know it's the blue light that's really responsible for the plant to be able to perceive what to do and how to regulate it. So in the morning, when the sun comes up and hits a plant, it's the blue quality of light that strikes the leaves and the stomata and the guard cells, and tells the guard cells, go ahead and open up. It's sunrise. We can start doing photosynthesis. All right, so that blue quality of light. So here, just some very simple experiments again, where they worked with those baby seedlings that have that coleoptile sheath tissue that's protecting the leaves. Anyways, the coleoptile seedlings are just very flexible. You can get a quick response. And so what they did was they were just shining those baby seedlings with different qualities of light and seeing if they could get the phototropism to work. And it really only worked when blue quality light. If they didn't have blue quality, just had red, for example, the plant just didn't respond to that unidirectional light and did not do the phototropism. So they've been playing with this now moving forward. This was discovered quite a while ago, but they've been looking at this for all kinds of plant behavior, like what qualities of the sun, what light energy are they really using in order to assess what's going on and then respond appropriately. So another idea that they've been looking at is this photoperiodism, is the fancy word, but basically it's studying how does a plant know, day versus night, and then use that information to decide when to flower, which obviously pretty important for the plant, because if the plant is kicking into that part of their life where they're forming flowers, that means they're ready to do sexual reproduction, get their seeds made, which is vital because that's how they pass on their genes into the next generation. And so pretty important that they get this right and don't flower at the wrong time of year. And so they're constantly assessing how long am I in the day versus how long am I in the night period. And so they categorize plants as they're studying this and figuring this out. You have these different categories that plants will fit into. So for example, there's short day plants. Another way to say that would be they're long night plants. So either way to say that is fine, but we tend to go with daylight. So short day plants. So what does that mean? It means that they're not going to flower unless there's some day length. They have to be under a critical day length. And I'll show you an image, we'll work through it to have this make more sense. So a short day plant will not flower unless they're below a certain critical time period, a shorter day. And then a long day is just the opposite. They're not going to flower unless they have periods of light that are longer than some critical time stretch, okay? So short day plants, I have in the notes section, cannabis supposedly fits into the short day plant idea. Ragweed fits into the short day. And then long day plants. What are long day plants? Do I have that in my notes? Oh, there it is. Beets, lettuce, potatoes, spinach, beet. Okay, so in the notes section, I have some examples. And you can find this out usually by Googling a reputable resource. You can find out if you're interested in a certain plant, whether or not they are short day, long day, or, well, here's poinsettia. And so they're a short day plant. They will only flower if their critical time is 8.5 hours. So if they've been exposed to hours below that, then they're going to flower. If they accidentally or somehow get exposed to greater than 10 hours or greater than 8.5, this one on the right was exposed to 10 hours, and then it won't flower. So I think that poinsettias are big business. Every year around the holiday, Thanksgiving, Christmas holidays, you see poinsettias everywhere for sale. It's big business. And so you better get this right and time it so that you have your plants in full bloom so that you can sell them and make a good profit during that time frame of the year. By the way, when you look at a poinsettia plant, all those bright red, those are not those are not flower petals. They're modified leaves. Okay, so the fancy word for that is bract. If someone says, oh, look at the bracts on that poinsettia, they're thinking about all the red modified leaves. So if you have modified leaves right next to the flower that take on a colorful something other than green, then we usually refer to them as bracts. So the actual flowers are these tiny little things that you see in the middle of all those bracts. So, but it's very impressive. We like the looks of it. Most people really do find poinsettias appealing, and so it's big business. So here you see a greenhouse just chock full of poinsettia in bloom. So they must have been receiving light lengths that were below 8.5 hours. I was once listening to a podcast where a greenhouse or a business owner, that's one of his primary plants was poinsettias, and he had hired a new greenhouse manager that came in during the evening and turned on the light to do something. And that whole greenhouse, all the poinsettias refused to flower in time. And when the business owner found out that his new greenhouse manager made that mistake, the podcast interviewer said, "Well, I bet you fired that person, didn't you?" And the business owner said, "Actually, no, because that person felt so bad that that happened. And he was so upset about it. He would never forget that. He will never repeat that mistake again." And so I gave kudos to the business owner. It's like, that's smart thinking. You know, if that person felt badly enough that they made that kind of mistake, yeah, you know, they won't make that mistake again. So critical that you get this right if you need to have your plants in bloom in order to make a profit. So there's also groups of plants that are called intermediate day plants. They're not going to flower if it's too short or too long in terms of how much light they're getting. And then there's some plants that we just say they're day neutral because they don't seem to really care. And these are often plants that are located along the equator, where they get kind of even Steven day-night rhythms pretty consistently throughout the year. And so day neutral plants, tropical plants, and plants adapted to more tropical conditions tend to fall into that category. So this whole photoperiodism idea is kind of thinking about how does a plant decide when to get into sexual reproduction, which for angiosperms is the idea of formation of flowers. And you may have noticed as you have been living your life to date, when you're out hiking around at different parts of the, let's say the spring, summer, fall, especially around here, you will see that some plants are flowering in the spring, but then some plants don't flower until it's more in the fall. And so again, it's due to their particular life strategy of, you know, how quickly can they make flowers and set seed. And they have to pay attention to the day-night length going on throughout the whole year to make that decision. And so, of course, from our perspective, we are very interested in how does the plant decide to flower, because so much of our food is based on the plant's going to flower in order to set seed and make the fruit that then we harvest and feed to billions of humans. And so big research, lots of dollars going into this. And so what they've found to date, one of the big players in figuring this out in terms of day-night rhythm is this photoreceptor that we call phytochromes. Phytochromes. So these are essentially, it falls into the category of a photoreceptor. Plants have many different types of photoreceptors. This is a pigment complex that will absorb certain qualities of light, and then the plant can actually assess what are my phytochromes doing, what are they like, and then based on that, they figure out what to do in terms of flowering. So what they're doing, and they think that phytochromes are mostly located in the plant body in meristematic tissues, but there's also been suggestion that they're also located in the leaf buds that you see, you know, sprouting in the spring when the leaves finally start to leaf out. It's the leaf buds. They think that there's also phytochromes there as well, which kind of makes sense because And the leaf buds, they don't want to bud out and form new leaves in the middle of winter. So they need a way to be able to assess what's going on with the light variation. So this is an interesting photoreceptor. There's two forms of it. There's the so-called piece of R form, and that form, the R stands for red light. So it just means that that version of phytochrome, its job is to absorb red light. That's what it does. And then there's another version of the phytochrome that's called P sub FR, and FR stands for far red. So its job, what it's designed to do, is absorb far red light. Now, the interesting thing is when, let's say, the P sub R form gets hit with red light, what it does is it actually shapeshifts into the P sub FR form. So we call this a shape-shifting photoreceptor. It actually physically changes shape. So, and the same goes with the P sub FR. When it absorbs far red light, it'll shapeshift into the P sub R form. So this picture that we're seeing is trying to get that point across, where here on the far left of the picture, you see P sub R. And you see here, if you follow the arrow on top, it's saying that if the P sub R absorbs red light, it converts it physically, it'll shapeshift into this P sub FR form. Then, if you have that form of the phytochrome and you hit it with far red light, it will shapeshift into the P sub R form. So it's a really interesting system here. It's pretty simple in the sense of it's just this physical shift in its overall shape that is helping the plant determine how many hours am I in the light versus how many hours am I in the dark. And so what the plant is actually doing is it's assessing, well, how much P sub R do I have versus P sub FR in my tissues? It's always looking at that ratio and assessing amounts of P sub R, amounts of PFR, and that will tell the plant how many hours it's been in the light versus the dark. So let me show you this other picture. This is not from your book. It's actually from my plant physiology textbook, but I like the idea that it's showing this physical shift. All right, so it's a shape-shifting photoreceptor. So here you see the so-called P sub R form. It's kind of bent down like that. And then if you hit the P sub R form with red light, it changes into a more Y-shaped form. And then they tend to say that the P sub FR form is active. What they mean by that is that it's typically the P sub FR form that actually will move into the nucleus and start impacting gene expression. So it'll tell the DNA, I want this gene turned on or this one turned off. And so that's why they say it's an active form, because it actively goes into the nucleus and starts affecting gene expression so that the plant can respond to this day or night light. Okay, so try to keep this in your head. It's a shape-shifting thing going on here. So the P sub R form, if it gets hit with red light, it shapeshifts into the P sub FR form, and that's the active form. And then if that form gets hit with far red light, it shifts back to the P sub R. And yes, the P sub FR is active in the sense that it directly will interact with the DNA and alter gene expression. But the plant is also assessing how much P sub R do I have. It's really assessing both, how much P sub R, how much P sub FR, and that is helping regulate its behavior to its environment. So probably the best studied impact that these phytochromes is on flowering. They've been studying this very closely, like how do phytochromes work to make, get the whole flowering process going. in these different plants. So let's take a look at that and just give you some examples here. So this is a plant that's considered a long day plant. So that means so this dashed line that you see in this donut, this first donut pie chart thing, you see that dashed line? That's the critical length. And so if the plant, this long day plant, is in sunlight longer than that critical period, it will flower. If this plant is too short of a day length, it didn't make it past the critical time period, it got less light than that, it will not flower if it's a long day plant. So that's how you interpret these kind of donut pie charts here. So they started doing this with all kinds of different plants, looking at, how do these, are they short day plants, long day plants, day neutral, intermediate, what's going on here? And so they have collected a lot of that data. But during this experimentation process, they were having some fun with this. So I'd like to just show you this idea here. So the first two donut pie grass we already looked at. Now, this is interesting. This third one here. So it's a long day plant again. They did not give it enough daylight. So if you just think about that, you would say, well, then it shouldn't flower because it did not go past that critical time period. So it shouldn't flower. But look what happened here. So they set up the experiment. They gave the plant a fairly short light period. It was, shorter than the critical period. But then they came in at night and they turned the light on for like 10 seconds and then turned it off. So this red light, they hit the plant with red light. And then they watched what happened. And this plant decided to flower. And they're like, what? So then they did an experiment again where they set it up just like what we just talked about. They came in the night, hit it with red light, and then like a half hour later, came back in, hit it with far red light. And when they did that and waited to see what would happen, the plant decided not to flower. So basically and then they've done all sorts of these experiments with all kinds of plants. And you can keep doing it here. You can see they got even fancier with hitting it with different versions of light red or far red. But basically what you can see in these last 3 examples or 4 examples here is that the plant is paying attention to the last quality of light it was exposed to. And so here with the red, the third one, with it getting hit with red light, it's as if the plant experienced light all the way up to that red light exposure. And so it thought it had surpassed the critical day length and it flowered. So it's important to think about the idea of red light. When sunlight is striking a plant, it's really paying attention to red light for more than one reason, but the phytochromes in particular. And so if a plant is in full sun, it will be getting much more use of red light. Far red light, it doesn't care so much about, so to speak, and will often let that pass through. It won't really absorb it. All right, and so I'll talk a little bit more about that. But so this red light, it's as if the plant goes, oh, I'm in sunlight and I made it's past the critical day length, so I must have been in light this whole time. I'm going to flower. But then you come back and you hit it with far red after a red light exposure, and now the plant's like, oh, far red, that's dark. I'm going to be going into the dark, and it won't flower. So it's going back also to this idea of what's happening here. So when this long day plant got hit with that far red light, it shifted its phytochromes into the piece of bar. And when a plant has a lot of piece of bar in its body, it's saying, I'm in the dark. It's one clue that it has. If there's lots of piece of bar, It's either being shaded out or is in dark. If it's getting hit with a lot of red light, that's telling the plant, oh, I'm in the sun. I'm getting hit with light. And it's also converting the piece of R to the piece of FR active form. So the plant, this long day plant that we see with this experiment, it's always assessing how much piece of R, how much piece of FR, what's my ratio, and then based on that, it will decide whether or not it's met that critical day length and what to do. So with a long day plant, it has to be longer than that critical day length in order to flower. And so if it's getting lots of red light, that means most of its phytochrome is in the piece of FR in its body. And that's telling the plant, oh, I'm in light, I'm in light. Because in order to have piece of FR, that means the piece of R form had to get hit with red light. So really think about the red light, far red light idea. Let me show you one other idea here to kind of help with this idea. So think about an ecosystem, a forest ecosystem, where you have plants that are in different layers of the canopy. So you have the uppermost canopy where they get full on sun. They're getting all the sun directly onto their leaves. But then you have the lower canopies that will get dappled sunlight. And then you have the understory, more of the ground level plants that are getting a decent amount of shade and maybe some dappling of the sunlight. So what you need to think about here is that let me go back to this. If you have sunlight energy hitting that uppermost canopy, those plants will primarily absorb red light. The phytochromes are going to be absorbing red light. The chloroplast pigments will be absorbing red light. Plants love red light for more than one reason. Yes, they use it for photosynthesis, which we'll talk about, but they also are doing it for this phytochrome thing. So that means the upper story trees, if they're getting hit with full-on sunlight, the red light's hitting their body, and so they're going to have a lot of piece of FR in their body. And that's telling them, oh, I'm in sunlight. And maybe they'll decide to flower, depending on if they're a long day plant or not, or what time of year it is. But if you're an understory plant, you're not going to get as much red light as the upper story. They've paused a lot of it. So they took their share of it, which means there's less of it going through to the understory. So if you're an understory plant, you're not getting as much red light. And instead, you're getting more far red light. So if you're an understory plant and you're getting a good dose of far red and just a little bit of red, what's happening is if you're getting that far red light as an understory plant, that means most of your phytochromes are going to be in the piece of R form. And that's also telling you important information. So if you're an understory plant, you're like, gee, I have a lot of piece of R. I must be getting shaded out. And depending on what type of plant you are, you might have to change your behavior. Now, if you're an understory plant and you're adapted to that, it might be fine. You might go, oh, I'm getting a lot of piece of R here in my body, but that's all right. I'm shade adapted. I'm good with that. But if you're a plant that's a full-on sun-loving plant and you like full sun, if you're in that situation where you're getting a buildup of piece of R in your body, you're going to go, uh-oh, I'm getting too much piece of R here. That means I'm getting shaded out to some extent. I love sun, so I got to do something. I got to change my behavior. So if you were a plant rooted in place, you can't walk out from the shade. You're stuck. So what could you try to do to get access to better sun? Yeah, grow out, right? You might do that. You might say, especially if you're a tree, that's the strategy they do. They'll grow out longer so that all their leaves anyways are in the sun. You could grow taller. You think you're getting shaded out and go, well, I'll just get taller than the person above me. I'll get access to sun. So growing either lateral or upward, it would be a good strategy if you're a full-on sun-loving plant and you sense, oh, I'm getting shaded. All right, so they're using the information of both. They're looking at how much piece of R, how much piece of FR, and they're looking at that ratio. And based on that, that's telling them if they're in the light or in the dark. And then depending on the plant, it might be totally fine. It might be, oh, I need to do something about this. Okay. So this was a big deal when it was discovered. This shape-shifting idea was quite fantastic. They had never discovered anything like that before in plants. And so, and I'm quite happy to know that one of the people that discovered the shape of phytochrome was a former NMU botany student. And so he graduated quite a while ago, but he went on for his PhD at Madison and was first author in a publication into the Science Journal, the journal Science, which is like you get published in Science, that's a huge feather in your cap, so to speak. And so he did really nice work on looking at the overall formation of that photoreceptor. So, okay, this is a little bit complex. Are there questions right now on how this works? Yes. Well, in a natural setting, perhaps, yeah. But if you have a controlled, environment where you can control all of that, then it doesn't really matter. But when I first learned this, I'm like, well, what about moonlight? Like moonlight at night, wouldn't that have an impact? Because a bright moonlit night, it can be very bright, and they even show that moonlight can impact this, how their PR piece of FR. So that's kind of interesting. And then I also worried about, well, what about my outdoor lights around my house? And I tend not to have bright lights around my house. I just don't like that. But a lot of people really want, and I thought, I wonder if that, and it does, it can impact your plants in your yard. So. Yeah, so if they're a shade loving plant, if that's what they're adapted to, then they're fine with a lot of piece of R in their body. That's telling them I'm in the shade. That's all right. That's what I'm adapted to. I'm fine. Yeah. Yes. So you really, if you're working in a greenhouse situation and you're trying to get those plants to flower, the business owner should be paying attention to, are they long day, short day, intermediate, day neutral? They should know that and be able to regulate their their lighting regime based on this information. So, oh yeah, yeah, it'd be good to like have a generated list that you could like hand off to customers so that they know. Yes. It's just the Photochromes protein that to right. That's the natural selection processes. They just adapted that phototropin protein. That's the one that is delegated to absorb light, and that is something that, oh, it's not raining now. I need to regulate my guard sign. So exactly why? I don't know that they know. So there's lots of different photoreceptors that a plant has. Phytochrome is probably the most famous, and one of the first to be discovered. And it's also famous in that it's one of the first to be discovered that can shapeshift like this. But there's a series of other pigments, photoreceptors that can absorb different qualities. Even ultraviolet, there's some photoreceptors that can pick up that they're getting hit with ultraviolet. And of course, as we know, ultraviolet can be dangerous. So they need to understand that information too and respond to it appropriately. Of course, they can't go into the shade if they're getting hit with ultraviolet like we can. So they have to kick into defense responses, essentially. They have to try to get their repair mechanisms up and running because ultraviolet can damage them just like ultraviolet can damage us. So this will not be on, excuse me, this will not be on this exam on Thursday because I think it takes you a handful of days to work through this. And I will be asking questions on the second exam about how phytochromes work, what's going on with the ratio, what is that telling the plant. So please spend some time. I know you're not going to really focus on it in the next 2 days perhaps, but as soon as you get done taking the exam on Thursday, go back to this lecture and please study and look at this more than once. Because I know that I grappled with it when I was first learning it. It takes a little bit of time, at least it did for me, to have it sink in. So talking it out loud, talking through it, drawing this, I would draw this over and over and over. I would use this visual idea to help me understand what's going on. And then working through some scenarios, like what happens with, light versus dark critical periods. Okay. So phytochromes are really, really important for flowering. It helps determine the plant is assessing, day length versus night length. But it can also, the more they study it, they're finding other things that phytochromes can do for the plant. So overall plant development, they know that phytochromes are responsible for part of that. They're not the only thing, but they play a role in overall plant development. Plastids, getting plastids to become the different kinds of plasmids. So, you have chloroplasts, you have chromoplasts, you have leucoplasts. And so all of that. Anthocyanins are pigments. You learned about those in lab when you did the cabbage experiment and dyed some wool yarn. And then shading ideas. A plant needs to know if they're in a more natural setting. Certainly if they're sun-loving plants, they need to figure out if they're in the sun or in the shade. And so detecting of shading is really important. But even if you're growing plants for living out in a field like corn, that's all you're growing is corn. They have done some nice studies on that too, that individual leaves of nearby plants, they will shade each other. It's highly likely you're going to get some partial shading of one corn plant neighboring another. But they've actually showed that corn plants can actually orient their leaves so that they're not shading each other too much. And so they've done some really sophisticated studies showing how neighboring plants will actually kind of go out of their way to stay out of each other's sunlight path as much as they can. And some of these studies are really, really cool. So really important to be able to assess sun, dark, day, night. Am I getting shaded? So these photoreceptors, again, the way they tend to work, it seems like it's this shape-shifting idea where once they absorb the quality of light that they're adapted to, that causes a shape shift in their overall morphology. And then that is what is telling the plant some important information. And so we often refer to that as like when the phytochrome gets into the piece of FR form and then it goes into the nucleus. That's like we call that a signal cascade. That the phytochromes are involved in kicking off the signal cascade because the signal has to then get into the nucleus and alter gene expression in order to get the plant to behave differently. Okay. So the fancy word is it's doing a photo response. It's responding to that quality of light. So photoreceptors, basically what they're doing is they're absorbing light energy, but they're not using it for photosynthesis. They're just using it as a signal that they can transmit to the nucleus of the DNA. And so it's a different process than what you think about with photosynthesis. So again, this shape-shifting idea. And so there's been many different photoreceptors that have been discovered since then, and I'm sure they'll continue to discover more. So it's this idea of acting as a kind of kicking off the signal cascade process, where you get this information sent to the nucleus and then to the DNA. So with more and more experimentation that they've been doing with plants, how do they respond to their environment? How do they behave? There is a quote that I put here from Dr. Stefano Mancuso, who's I think he would describe himself as a plant neurobiologist. I know that sounds strange, but there is a big burgeoning field of research looking at plant behavior and that they can act with intentionality. And so there's a lot of interesting research coming out. I talk a lot more about that in my advanced botany class when I teach that. But this quote, I think, is awesome. To survive damage, decentralize. So modular and distributed design. So what they're saying here is, well, I'll read the quote. Plants distribute over their entire body the functions that animals concentrate in specific organs. Decentralization is the key. We have discovered that plants breathe with their whole body, see with their whole body, feel with their whole body, and evaluate with their whole body. Spreading each function over the entire organism as much as possible is the only way to survive predation and damage. And plants can do it so well that they can even withstand removal of much of their body without losing functionality. I mean, it really is a phenomenal way to live your life. That decentralization, very, very different than how we live our lives. You know, if someone chops our head off, we're done. You do that to a plant, and they're like, all right, I'll just, you know, my axillary buds will kick in. All right. All right. It's really amazing. In the fall, if you cut back your rose bushes, I mean, you can get so aggressive and cut them way back. And in the spring, they're like, yeah, I'm fine. I'll just grow. So plants are amazingly plastic in that way. The plasticity of their body to be able to, and it's true. I mean, they have so many touch receptors on their whole body. When you stand next to your houseplant, it knows you're there. It can sense you. And so there is so much more going on in the plants than we ever gave them credit for. And I'm so excited to see the research is finally catching up and starting to reveal these mysteries. It's really fascinating. Another idea I just wanted to put into your thoughts also is this totipotency idea. This is also phenomenal because you can take just a handful of cells from a plant body and clone that plant through tissue culture technique. And so, you know, we can't do that if we just take a few of our finger cells. We can't put that into a tissue culture and grow a new finger. At least not right now, we can't do that. So you can take just a handful of cells from a plant and clone it into a full plant using the proper tissue culture techniques, the proper ratio of hormones and nutrients. And so we do this experiment traditionally in plant phys class. In my new class that I'll teach in the fall, the applied plant science will also dabble with this concept as well. So again, just remarkable ability. And again, you know, if you think about it, they have to be able to survive predation. There's all kinds of things coming along and chewing on them, and they can't run away from it. They can't shoot it or kill it or fight it off with any kind of physical means. They have to do it through very sophisticated chemical means. And so it makes sense that they can get chopped down and chewed down to almost nothing, and yet they can bounce back. So wonderful plants. All right, we'll stop there. And remember, I will hold lecture on Thursday, but that content will not be on the exam either.